Okay. Before we get started, just want to remind you, this coming weekend is the first midterm, right? And if you're one of those people that want your whole weekend free, you can take it anytime between Friday at noon and before midnight Sunday, right? So you decide when to take it. Once you start it, you only got 90 minutes to answer 100 multiple choice questions. And Tone has posted on the D12 site a study guide, if that's helpful to you. And what else? And you have to be finished by midnight Sunday night, okay? If you're one of those people that want to use your Friday for something else, then you've got the whole week. So how's everyone feeling about that format? Are the people that would rather have tests in class with scantrons and all that? Are the people that would rather have the tests be synchronous, where everyone takes it during class periods? Okay. It's an experiment. We'll see what happens, okay? Other questions about the format of the course? See, lots of people got 3s on their first perusal assignment. Remember, that doesn't count towards the 20, but if you got a 3 on the first assignment, then you know how to get them in future assignments. And I had to do a little more shuffling around of people because I had a couple of people show up that hadn't been there before. Is there anyone else enrolled in the class who has not yet purchased a perusal copy and therefore had a chance to... Anyone? Okay. So I think we're set on this, and that'll be interesting. I will apologize ahead of time. Through sheer carelessness, I did not bring my stash of blow pops with me today. Of course, we're still going to have a Kahoot, but I'm embarrassed to say your reward will be, well, I'll owe you. I'll bring the things later. Doing okay? Okay. In chapter 1, we realized how to build a really good metazoan, a really big one. You got to have cells constantly communicating with each other. And in chapter 2, we figured out by having a few anchor points, you could have that communication between cells, each of them dictating what genes the cells will express, and therefore what role they'll take in the adult animal. By just having a few relatively simple rules like that, you end up building the entire organism with a body plan, one that works. And by works, of course, I mean it can reproduce itself. And now we're talking about what happens later when now you've got to make a whole bunch of neurons to populate this nervous system that we're going to care so much about. And we were talking about how it turns out that neurons never divide. Any neuron that's dividing is something's gone terribly wrong and it's a cancerous cell, right? And so neurons themselves never divide. So neurons do not give birth to other neurons. Rather, neuroblasts divide, and at a terminal division, one offspring could become a neuron, or it could become a glial cell. And we saw in those studies in vertebrates how you could mark a dividing cell near its last few divisions. And you could see that the fate of its offspring, children, grandchildren, great-grandchildren, could take on just about any fate. And that tells you that in vertebrates, at least, this ability of the cells to listen to their neighbors, to decide what particular role to take on, stays with them very late. It's a very late decision. Another way to put that is they remain plastic or pluripotent rather late in the process. All the indications are, even up that last division, in vertebrates at least, unlike C. elegans, in that last division, the fate of each of the daughter cells is pretty much up for grabs. With me so far? And that fact that neurons don't divide, but that the precursors divide, mean that we have one way of keeping track of development by birth dating cells. Giving the animal some DNA-specific precursor, something that's only taken up in DNA, like thymidine, or a synthetic molecule like bromodeoxyuridine, never occurred in nature before, but it is enough like thymidine that dividing cells can't tell the difference and use it and incorporate it in their DNA. But it's enough different from thymidine that we can make an antibody that will let us see it And so we can talk about the birth date of a cell based on when the marker was available to it. And we can look anytime after that last division, we can look and see what fate it takes on. And so here's just a reminder about how immunohistochemistry or immunocytochemistry for bromodeoxyuridine would work. You injected the animal when, say, it was in the 12th day of gestation, fetal development. You let the animal grow up, you kill the animal, take out the brain, slice it in really thin sections. And now in those sections, any neurons that were undergoing their last or near last mitosis back in the 12th day of gestation, they'll be marked and we'll be able to identify. And what's cool about bromodeoxyuridine is that identification is pretty fast. So you take the brain section, you float it in solution, you have in that solution antibodies that are specific for bromodeoxyuridine. You give them a chance to bind to any BrdU that's in there, and then you rinse off the excess, and you take through a chemical reaction that will let you see where the antibodies are. And lo and behold, you'll see a cell somewhere, in this case in the hippocampus, where there's bromodeoxyuridine in its nucleus. And you might be wondering, well, is that a glial cell or is that a neuron? And then there are other immunocytochemistry things you can do to look at that, where you can say, expose the animal to some task that will induce an immediate early gene expression like CFOS, right, which is something that neurons do. And then you can see both the BrdU in the nucleus and the CFOS in the cytoplasm in the same cell. And so that cell that was activated by whatever I did to the animal just before I killed it, it was born way back in the 12th day of gestation. How you doing? This use of birth dating revealed a really important thing about the development of the cerebral cortex. So several people had remarked or noticed in lots of preparations, including painstaking electron microscopy reconstructions, had noticed that in the developing cortex, there comes a time when there are these glial cells marked with GFAP that are still holding on to the peel surface and the ventricular surface. So they've been stretched out incredibly thin. And we recognize them because they express GFAP on their membrane. And people had noticed that there seemed to be cells, almost amoeboid-like, that were wrapped around these processes. And eventually it became clear that these cells are neurons to be, that they underwent a final division down here in the ventricular zone, and then began climbing up the radial glia to get to the top, and eventually populate all 6 layers of the cerebral cortex. And it turns out that there's a very regular relationship. Oh, and I'll just remind you, this is sort of a generic slide, that the 6 different layers of cerebral cortex, which defines neocortex, which is important in mammals, for example, the 6 layers happen to be numbered with Roman numerals, 1 for the outermost, 6 for the innermost. The outermost layer, so-called molecular layer, or lamina 1 in the spinal cords, the outermost layer consists of almost no cell bodies. It is almost exclusively dendrites and axons in glial cells and synapses between the two. And this is where a lot of the processing in the cortex takes place, is out there in that molecular layer. And then there's the so-called external granule cell layer of the cortex. Confusing, because we're going to run into an external granule cell layer in the cerebellum, which has nothing to do with this. All they have in common is the granule cells, meaning about the size of grain of sand. You can blame Ramon Cajal for that. And so all it means is that they're small. Doesn't say anything else about their fate or their function. Just they're small and external because they're towards the outside. And then the other neurons that Ramon Cajal made famous are the so-called pyramidal cells, which in the cortex are among the very largest cells that you'll see there. And they're called pyramidal cells because their cell bodies are shaped like pyramids, but they have this very characteristic trait that they send one axon straight up to the surface, towards the surface, and several other axons that are branched out below. So we call that axon, that dendrite that's going straight up, the apical dendrite. And then they also have basal dendrites. And remember, the dendrites are where synapses are going to come in. And then these neurons, like all neurons in the cortex, they send their axons either to other layers of the cortex, or eventually their axons might pass all the way through into the white matter, where they'll be myelinated and go to other places. And for the most part, cortex talks to cortex. There's some interesting exceptions. After the layer 3, where the some pyramidal cells is, then there's another layer, so-called layer 4, internal granule cell layer. Again, so these cells tend to be small. They're not as big as the pyramidal cells in layers 3 and 5. And it'll be layer 4, where any input to this bit of cortex always comes in synapses in layer 4. It's really striking how regular this pattern of neuroanatomy is in the cortex, right? It's going to be primarily the pyramidal neurons that send the axons out of cortex, and any information coming into cortex is going to synapse first on those granule cells in layer 4. That'll be important for some of the later studies we look at. And we won't be talking about this a lot, but it also turns out that almost all of the synaptic connectivity that's going on in the cortex is all happening in tiny little columns of cortex. So imagine this bit of cortex is a little silo of cortex and a column. And within that column, the neurons are talking almost exclusively with one another, except for the inputs that come to layer 4 and the outputs that go out from layers 3 and 5. And your cortex consists of trillions of these little columns, and there's pretty good reason to believe that each little column is analyzing a particular little problem. Taking in information, maybe sensory information, maybe information from other parts of the cortex, analyzing information, and sending out an axon to tell you to do something. And then finally, there's a catch-all layer at the bottom, layer 6, so-called multiform layer, because there's no particular pattern of the sort of neurons you see there. How's that? Good. Yeah. Laura.
You said that each column is kind of like the response to fertilizing one column, like the issue. So if you damage that region, you're kind of like.
Yeah. So it's interesting that, so if you could find the region that helps you understand how to integrate, how to do integrals in calculus. If I were to kill that one little column, would you be helpless? And the answer is no, because for a problem, any problem that we can identify and put a name to, it's going to involve lots of columns. And so there's a lot of redundancy there. Yeah, good.
I can stay on your console.
Oh, I've got the different stains up there, but never mind what it's staining. The different stains, the only ones we'll talk about is the Golgi stain, which shows just a few neurons, but shows them very completely, and the Nissl stain, which stains all cell bodies that have any RNA in them, because that's what Nissl stains stain, is RNA. And those are far and away the most common. Good questions, all of them. So I told you there's a very regular pattern of the birth dates of cells and how neurons end up in cortex. And to introduce that, we need to talk about what's going to happen during development. So at first, there's the so-called cortical preplate, where all the cell bodies are going to be, and there's the intermediate zone, and then there's the ventricular zone. And so all the division's happening in the ventricular zone, and then the cells come up out of the ventricular zone and pass the intermediate zone and start making the so-called cortical plate. Just a catch-all phrase for this is where the cortex is going to be. And then there comes a point when the first cells that come in, well, eventually what we're going to see is the first cells that come in are going to form layer 6 and are going to avoid the marginal zone. And the next cells that come in are going to make layer 5. And the next cells that come in are going to make layer 4, then 3, and eventually 2. So that by the time it's over, you've got this sandwich of the outermost layer, layer 1, going to be the molecular zone, almost no neurons in it. And it is sandwiched beneath it is the white matter, the previous intermediate zone. And this development of layers is very much related to birth date. And so let's look at that. In a landmark experiment, the great neuroscientist Pascal Rakish at Yale did some truly heroic experiments where he injected tritiated thymidine into developing macaque monkeys at different stages of development, of gestation, right? So he injected some on the 30th day of gestation, some on the 40th, 50th, etc., right? And he let the animals grow up, and he killed them, and he processed the tissue, and he knew that any cells that had the tritiated thymidine in their nuclei were those that had been undergoing their near final division way back here when he provided the thymidine. Everyone get the idea? And when he compared all these adult animals that received the thymidine at different ages, he found something very interesting. First of all, the very oldest cells, the ones who underwent their last mitosis earliest in development, there were hardly any of them. Some of them were in the white matter, and some of them were in that molecular layer. But the important thing is he had to look really hard to find any, because there are hardly any neurons out there. Remember, the outer layer, layer 4, is mostly neuropil, and the inner layer, the intermediate zone, that's white matter. Hardly any neurons there. And why does it look so white? White matter. What makes it look so white?
No.
Pardon? Myelin. And why is myelin white? Pardon? Because fat would have been fine. They're made of fats, lipids, right? You know how Crisco is white? That's why myelin's white. Same principle. Okay, doing so far so good. And what Rakish found is that in all these layers in between, 2 to 6, where almost all the neurons for the cortex are going to come from, the oldest of those were in layer 6, and then layer 5 consisted of cells that were born a little bit later, and layer 4 was cells born a little bit later than that, and later than that, and that layer 2 had the newest neurons there. So there's this inside-out development of the cortex where the oldest neurons, by which I mean the ones who has the earliest birth date, by which I mean the ones who kicked out of mitosis earliest in development, they're populating layer 6, and layer 5 consists of cells that are a little younger than that, 4 younger than that, etc. This inside-out development. Keep in mind, it could have been differently, and no one predicted this would be the pattern, right? It could have been different. It could have been that the ventricular zone keeps churning out cells, and they migrate up, and you can imagine each one. Well, here's a nice little place for me. This isn't popular. I'll go here, right? But that's not how it is. They go right past their older siblings and establish a new layer outside that. So far, so good? Good. All right.
So I guess the first layers and like an section to that.
Yep.
They like 30 their responsibility.
Right.
Like not the
What's in the middle?
first day that the
Oh, during the first, remember in the first days of development, that doesn't exist. The plate hasn't happened yet. So
up there and the first like
So these cells, right? So early in development, there is no layer 6, there is no layer 5. They haven't been formed yet, right? And so what happens as the neurons form those layers, then the older cells get wedged apart so that some of them end up in the outer layer and some get stranded in the inner layer, and there's hardly any of them in any case. By the time all that neurogenesis happens in the ventricular zone and the other 5 layers get populated, they're very much in the minority because they're not dividing, right? They stopped dividing a long time ago, back when the brain was tiny. Good. Oh, and importantly, if he did the injections, any of them after 110th day of gestation, he never saw any labeled neurons at all. So these monkeys, right, they're not going to be born until about 180 days of gestation. They've made all the neurons they're going to make, he said. We'll find exception to this later. In the middle of gestation. And that's where those come from. Questions about that? So this is where the majority of neocortical neurons come from the ventricular zone, with neurons that shinny straight up radial glia and get off once they get past any pre-existing layers. And this is where most of the billions of neurons in your head come from. We call the ventricular zone, another term we might call that is to call it a germinal zone. And this doesn't mean germinal as in germs. It means as germinal as in something that produces lots of different variety. Germs are called germs because they can proliferate. That's why that name got to them. The original meaning of germinal means like a germinal idea. It's really fruitful. It gives rise to lots of different things. So the ventricular zone is a germinal zone. There are also other cortical neurons come from some other germinal zones that are called the ganglionic eminences. If we look at a cross-section of developing brain, these ganglionic eminences line the interior ventricles. There are 2 in the front and 1 in the back, and they also are dividing like mad. And they tend to be born later than those cortical cells we talked about. And while the cortical neurons we talked about before, while they are going straight out the radial glia, climbing in gym class, they're shimmying up or shinnying up there. These cells are behaving more like Tarzan, going from radial glia to radial glia. They go across the radial glia and populate the cortex. And what they all have in common, the vast majority of these neurons are all going to eventually become tiny neurons that use GABA as a neurotransmitter. And I'll remind you that GABA is almost always an inhibitory neurotransmitter. And so you can see these latecomers as the ones that are going to put the brakes on various processes in the cortex and serve as an inhibitory influence. If something goes wrong, that you don't have that many GABA neurons or they're not that effective, this is when things like epilepsy can happen, right? When there's seizures, where there's overexcitation of the cortex and with bad results. So far, so good. And then it turns out, just in the last few years, we've learned that in humans, there is a very late population of neurons that are shinnying around after birth, starting before birth, but continuing after birth, where these neurons come from the anterior pole and start migrating up over what's going to be the corpus callosum, or migrating through the cingulate cortex. That strip of cortex is just above the corpus callosum. And then once they get in various places, spreading out and shinning their way up to the cortex overhead. These cells are also tend to be GABAergic. And what's interesting is these neurons are being generated and are pushing themselves into place after the baby is born. So extreme neuroplasticity. So far, no one's documented this happening in any other species. And you wonder if that innovation, that business of continuing to make neurons and fitting them in where needed so late in development, is one of the reasons why humans do calculus and most monkeys don't, right? It could be important. The other thing I'll point out to you to foreshadow what we'll see in chapter 10 is what's also interesting is that this level of plasticity is happening at a time when the infant is being tutored massively by the parents and other conspecific, right? People are talking to the baby, that people are moving the baby around, people are looking at the baby, it's getting a lot of social stimulation, and maybe that's important to make that social stimulation really set in and be effective. There are some places in the adult brain where neurogenesis continues throughout adulthood, despite what Rakesh reported in monkeys in that earlier report. Maria?
Yes, I have a question on both slide on the being not amino sensor. On the slide, it said like there's like the two steps of migration. One, and he to the cortex things. What is like the tangential?
Oh, by tangential, I mean, so they're not lined up with the radial glia, they're going across the radial glia. So they're taking a tangent. And pretty good evidence that they grab one radial glia, then let go of it and send out a process to grab the next radial glia and pull themselves along. So they really are all, it's pretty startling. They're all behaving like individual organisms, right? They're all little amoebas.
Are they also death rates the time?
No, not at this point. Not here. We'll see some cases where they do, but in this case, no. And these GABA neurons, they're all going to be interneurons. They're going to get synapses from some of the local neurons, and they're going to make synapses with local neurons. They are not going to be pyramidal cells. They're going to send their axons outside their little column of cortex. And that's probably important. Amy?
So you know what it is.
Yeah, great question. That's chapter 5. Wait, yeah. How do they know where to move to? Because they're sniffing around their environment constantly, picking up cues from, other cells. Oh, so that's how they know where to go. Other cells are telling them where to go.
Question about epilepsy. So if epilepsy is like when like the neurons immediately not evident or not, there's fully developed causing like the US inhibitory.
Yep.
So there's too much saturation in the brain. And if when the epilepsy could only happens when development?
No, because let's see. It is true that there are some children that may have seizures once in a while and they eventually grow out of it, right? And I didn't mean to suggest that most cases of epilepsy are a failure of GABAergic neurons to move about, but there are some that are that way. But of course, some seizures, persist into adulthood. They can be very, and there's certain things that can cause seizures that terrify parents, but are totally innocuous, like a really high fever. Sometimes when kids are really super tired, things like that, they may have a seizure, the parents panic, call the doctors, and they say, well, has it ever happened before? And they say, no, it probably never happened again, but call me if it does. And most of the time it doesn't, right? So seizures can happen to any, and most anti-seizure medication are drugs that are acting on GABA receptors to put the brakes on a little bit more. Well, they can only be effective then if there are GABAergic synapses that are normally inhibiting these things, right? Okay. When I was in graduate school, everyone knew that you were born with all the neurons you're ever going to have. Nobody knew about the late arc migration of neurons, et cetera. But it turns out that that was never true. And there was, and even when I was in graduate school, there was plenty of reports out there of neurogenesis happening later in life, even into adulthood. The first thing to tell you about these cases of neurogenesis is they are a small number of cells. They do not represent the vast majority of cells. And that's probably why they got overlooked for so long. But there are several places where a neurogenesis continues into adulthood, and they're interesting. One of them is in the olfactory system. So to remind you about the olfactory system neuroanatomy, the olfactory bulb in humans, it's kind of tiny and flimsy, looks like almost like a ribbon, but there is a bulb there full of neurons, and it lies at the base of the brain and over part of the skull that is underneath the nasal sinus. So air goes in your nostril here, and there's a surface of cells known as the olfactory mucosa, where there's lots of cells that are making mucus all the time to keep it from drying out. And in one part of the nasal passages up there, there's a part of the bone that has holes in it. And this is called the cribriform plate. Hard to pronounce because of those two R's, cribriform plate. It's kind of like saying February, right? It's not easy to do. But if you pronounce it that way, it'll be easier for you to spell it correctly. Not that I care less. Anyway, and so in the olfactory epithelium, there are cells who send dendrites into the epithelium to detect chemicals, odors. These are olfactory sensory neurons, and we'll call them OSN. And their dendrites stick out in the snot to detect any chemicals that land there. And they send their axons through these holes in the cribriform plate to enter the olfactory bulb and synapse there. These olfactory sensory neurons can be generated throughout life. You can make new ones. Sometimes people working in industry may get their nasal passages exposed to organic solvents that kill all their olfactory sensory neurons. They go home and they can't smell anything. We say they're anosmic because they can't detect any odors. But if they get away from the solvents, after a few months, their sense of smell comes back. The COVID pandemic, for many people, one of the symptoms of COVID was that they became anosmic, probably because the virus killed all those olfactory sensory neurons out there. But once they recovered from COVID, they generated new ones, even adults, and eventually most of them recovered their olfactory sense. It also sometimes happens that somebody has a real blow to the head, a really sharp blow to the head, and if it's just the right way, then the brain may move while the cribriform plate stays put, and so the olfactory bulb may break away from the cribriform plate and all those axons get broken. So now they can't smell because, and they too usually recover their sense of smell because they make new olfactory sensory neurons and they send their axons in and synapse. There's several peculiarities of the olfactory system I want to remark on now. One is that in the olfactory bulb, the neurons that are processing the olfactory information, they all form little globes, little globular areas that have a special name called the glomerulus. So each of these little spheres here that we're indicating consists of a bunch of neurons that are busy taking this olfactory sensory information and processing it, and eventually sending commands to the rest of the brain to the so-called mitral cells. So-called because they're shaped like a bishop's mite. That helps you. The other peculiarity of the olfactory system is these axons. This is the only sensory system that does not pass through the thalamus. The axons from the olfactory bulb, they go straight to prepiriform cortex, which is the primary olfactory cortex. And people have, speculated, well, maybe this is why olfactory senses are sometimes have some primacy. These tend to be people who read Proust and think that he's really important. But I don't know that olfactory sense has anyway, I'm skeptical of these ideas. I guess that's what I should say. But if so, then they say, oh, that's because it doesn't have to go through thalamus. But why would that make one modality primary over any other? What's wrong with thalamus? Okay. Where do these new neurons come from? They're right out there in the olfactory epithelium. So there's a cohort of cells out here that are not olfactory sensory neurons themselves, but they can divide. And sometimes when they divide, they kick out of mitosis and take on the fate of all olfactory sensory neurons and take over. Probably it happens when one of the neighboring cells dies, right? Even pre-industrial society, you sometimes run into chemicals that are toxic. And so they probably kill the neurons that got whiff of it and warned you about, oh, that's a bad odor, stay away from that. Another place where, yeah, good.
Well, and people you saw sterile olfactory neurons that had a they report that smell differently.
Oh, when things come back, it's a great question, and the answer is no. Cinnamon still smells like cinnamon, bacon still smells like bacon, all that stuff. And which means that the new olfactory sensory neurons, I didn't say this yet, but we know quite clearly that each of these glomeruli is processing information from a very select subset of olfactory sensory neurons that are all expressing the same chemical receptors to detect chemical. So somewhere in your olfactory bulb, there's a glomerulus, actually there are two, that are processing bacon. And another is processing Parmesan. And another is processing cinnamon, etc. And if you had any idea what the dimensions are of olfaction, you might ask, well, are they laid out in a orderly manner in the olfactory bulb? But we have no idea, because what are the dimensions of olfaction? I know what the dimensions of light are, right? Intensity, frequency. Sound, same, but, is bacon more something than cinnamon? Yeah.
It's like, smell stuff. Some people like it.
Yep.
And it's some that olfactory neuron.
So, and the great example is cilantro, where many people think it tastes like soap, and many others of us think it's wonderful. Mostly not to do with taste, but to do with the odor. And I know that's understood, but I don't know what the result is. It's a genetic thing, right? So if you're one of those people for whom it tastes like soap, I am so sorry. I apologize. You should have chosen your parents more carefully. Okay. Another place where adult neurogenesis happens is in a place in the lateral ventricle, in the anterior end of the lateral ventricle, where there are cells that continue to divide and go from the subventricular zone there, then start migrating in a pathway, like camels in a caravan. They start migrating, and they're constantly migrating rostrally to the olfactory bulb. And this is a puny process in humans who have such a tiny olfactory bulb. But if you're a rat, where look how big the olfactory bulb is compared to the rest of the brain. For there, it's pretty prominent and you can see it. So there's a continuous supply of cells that will become neurons that will go to the olfactory bulb, and they take on two very specific roles there. They either become granular neurons or they become periglomerular interneurons. I don't need you to remember that. They're color-coded here, so this is how you're supposed to know that they're there. And so they're continually being replaced through life as well. And they are being replaced. It's not that the olfactory bulb gets bigger and bigger and bigger as more of these cells go on, right? So the number of cells doesn't change much. So new cells are coming in. That must mean some other cells have gone away, have died. We'll talk about that process in Chapter 7. Oh, the other cool thing is that they're not holding on to radial glia or anything like that, but they are surrounded by these specialized astrocytes. So it almost looks like they're being herded, right? Everybody stay within the tube on the left side and the right, and then when you get the olfactory bulb, spread out and do your job. Other cool thing is that some of them are actually still dividing while they're shitting towards the olfactory bulb. The last place for continued adult neurogenesis is in the hippocampus. I'll remind you that the hippocampus, the hippocampal formation, consists of the hippocampus proper and here, and the dentate gyrus. So the structure of the hippocampal formation is quite complicated. I don't need you to know it, but I'll offer you a visual prompt that might help you, okay? So imagine that you've got a couple of rain gutters. You know what they look like, right? And the one that's facing, in this case, so I should say this is the midline. So one of these rain gutters, consider that the cell bodies in the hippocampus proper. One of these rain gutters is here, and the other rain gutter towards the lateral represents the cells of the dentate gyrus. So imagine these two rain gutters, one nested inside the other, which means that the dentate gyrus cells are always going to be medial to the hippocampus proper. And the two together are known as hippocampal formation. And just to make things hard now take those rain gutters and bend them both so that they form a C shape. And orient that so that the tips of the C are facing rostrally. And that's what the hippocampal formation, that's what the structure basically looks like. That's the three-dimensional nature of it. If that worked for you, great. If it didn't, you don't need to know that. And just offer in case you wrestled with hippocampus anatomy as I did. One of the places where neurogenesis continues throughout life is in the dentate gyrus of the hippocampal formation. Within the dentate gyrus, there is the neurons form a granule cell layer. Again, that just means they're small. And these are continually being replaced throughout life. The replacements are coming from the subgranular zone of the dentate gyrus. So here we're using a stain for double cortin, which is the gene that's only expressed in neurons shortly after they finish dividing. And I won't tell you how that got determined. Just take my word for it. And so you can see here in the granule cell layer, there are some neurons that are new, just got there. And they arise from cells that are dividing in the subgranular zone, who then just move up into the granule cell layer. You okay so far? And I'll let you look in the book to see how there was some controversy about adult neurogenesis, but my take on the field is now it is widely acknowledged and agreed that there is continued neurogenesis into adulthood in the rostral migratory stream, in the olfactory epithelium, and in the granule cell layer of the dentate gyrus. Okay? And I'll let you read in the book about why it became a controversy. Questions about that? Okay. Justin, right? Yeah.
Did you say the rostral stream also continues into adulthood?
Yes.
Is that it?
The rostral migratory stream, in rats at least, it continues throughout life.
The olfactory one, not the inhibitory.
Wait. These are the ones that are going to live in the olfactory bulb itself, not the olfactory sensory neurons. They stay out in the epithelium. Bring in blow pops for everyone. That's what we'll do. That's easiest. Okay. One more place where migration is important. Oh, and let me have the sheet. Yep. Thank you. Archaeologists will study these in future years. We'll put these straight into the archives. Okay, thank you.
Legal.
Perfect. Okay, so another place where migration is really important for the developing nervous system is in the peripheral nervous system. And the peripheral nervous system, we recall, are going to arise from not the neural tube, but from the neural crest. And it's really interesting how these cells are going to migrate around and do lots of different things. So, among other things, in the head end, the neural crest cells are the ones that are going to make those brachial arches that we talked about. And so some of them are going to contribute to, the throat, pharynx, stuff like that. And others of them are going to make the peripheral nervous system. Some of them are going to populate the cranial nerve ganglia, right? Collections of cell bodies outside the central nervous system who are either getting axons from the brain and then sending their axons to the body or vice versa. And those are all derived from neural crest cells, those cells that got left behind when the neural tube got formed. And in the spinal cord, that's where we can really keep track of neural crest cells, and where, remember, once the neural tube was formed, they kind of budded off separately and are kind of hanging out there between the neural tube and the ectoderm overhead. And in the spinal cord, there are 4 major pathways they can take. Some of the cells are going to migrate just a short ways and start populating out here between the neural tube and the cells that are going to make the rest of the body, which are formed here in a long tube that are called somites. And as we'll see, the somites extend along the neural tube and they're continuous, but the quality of the somites is not continuous. So some neural crest cells will come out here and stick around, and they'll make the dorsal root ganglia. To remind you, the dorsal root ganglia, those knobs on the dorsal roots outside the spinal cord proper, their cell bodies live in the dorsal root ganglia. They have long extensions. These are unipolar cells. Remember those, right, from way back then? These are about the only unipolar cells you've got. They send extensions, one into the spinal cord and one into the periphery. And so these are going to be, for example, all the sensory cells that have one end of their axon out in the periphery to detect touch, temperature, stuff like that, or maybe muscle size, muscle reaction. And then the action potentials generate in the periphery, go through into the dorsal root, and then into the spinal cord to synapse in the dorsal spinal cord. So they're going to be sensory neurons. Other cells migrate further out, and these neural crest cells are going to make the melanocytes. These are the cells that populate the skin and provide pigment. And that'll be important when we look at chick quail chimeras that people made, where it turns out, we know that some of those cells must be quail cells, quail neural crest, because there's a band of brown there. Others, and we'll talk about the dorsal ganglia cells quite a bit. Other cells migrate just below the neural tube and form the autonomic ganglia. So the sympathetic ganglia that run up and down alongside the spinal column, and the parasympathetic ganglia. And finally, some that we won't talk about much, some neural crest cells will stop in there and form the adrenal medulla, the inside of the adrenal gland, the part that secretes epinephrine and receives innervation from the sympathetic nervous system. Okay, so I laid on all that on you fast. We'll go over it again later, but I just want to tell you that the fate of these neural crest cells, just to preview what's going to happen, which of these fates they take depends entirely on what pathway they take. So the migration itself forms them to take on a particular task, and what sort of factors they run into on the way to their migration. And that's what we'll talk about to end up chapter 3. And that will be on the test. So I'm pretty sure I'll finish chapter 3 on Thursday. Whether I do or not, it's all fair game on the test. Okay? Questions? All right.
Yeah.
Off you go. Thanks for the fun.
Yeah.
Good ass.
Yeah.